Well, hello everyone, and thanks for joining our virtual journal club today. This is a journal club that we've been holding at Johns Hopkins for about 4 years now, and it actually started out as a microRNA journal club, and we still do that microRNA journal club with Mark Halushka and Sam Das. Um, and that's usually on the first Wednesday of each month. But then on the what is it, fourth Wednesday of each month, we have this extracellular vesicle journal club. And a lot of times is overlap between the microRNA papers and the EV papers. But now that we're all, or most of us, are home with COVID-19 issues and SARS-CoV-2, we thought that we'd just make this virtual. And since we're making it virtual, it doesn't hurt to just open it up to everybody. So no extra work there, and we're really excited about the response that we've had from the community. I just wanted to make a few notes before I hand it over to Yao Huang in my group. First of all, there are a few different efforts that are ongoing to bring some EV science to people during this tough time. And one of those is being led by Jan Lutfall and Karolina Sukmaji. I think I saw Jan's comment earlier, so welcome, Jan. So please watch Twitter and Facebook for more information on that lecture series. And then we would like to actually make this journal club maybe a more regular event. So without interfering at all with the microRNA journal club that's on the first Wednesday of each month, we might make this thing a weekly event on all the other Wednesdays of the month. So if anybody is interested in contributing to that, just drop us a note and let us know. So we don't know how long this is going to last. Many of you probably saw that the ISEV 2020 annual meeting has been postponed. We found out that the Gordon conference on EVs is canceled as well. That was going to be held in July. So it might be a good opportunity here to use some platforms like this to bring people together. So thanks again for joining. And I want to now introduce our presenter today. So Yao Huang has studied at the Southern Medical University in Guangzhou in China, where she was in the group of Lei Zheng. And she has been in my lab for some time as a visiting scholar. And the project that she's been working on, I think, was a very difficult project to start up in my group, and one that we were actually very skeptical about. So the idea of actually extracting EVs from tissues is one that kind of made us very nervous to start with. And I actually did a little 3-week visit to Andy Hill's lab, where I got to learn from him and from Laura Vella, who developed this technique and really applied some rigorous characterization to it. So I just want to mention that if you're interested in tissue extraction, take a look at their papers. Other groups are doing this. So Jan Lutfall's group with Rosella Crescitelli had a very nice paper recently on extracting from cancer tissues. And so I think there's a lot of interest in this area for numerous tissues, because this is how we really understand what EVs are doing. Their function. I mean, yes, they might have a function in biofluids, but a lot of their function is going to be exerted locally. So this preprint now is on bioRxiv. You can take a look at it, and Yao is going to take us through it. So thanks everybody, and thanks Yao for being willing to present at this inaugural virtual journal club.
Okay, thanks so much for Dr. Witwer's introduction, and I saw the greeting from all over the world, and welcome to our first virtual EV Journal Club. And I'm so excited to be the first one to present a paper by this way. So today we are going to present a paper actually from our group, and it's now online as a preprint and also under revision. So it would be great for us to discuss it with you to get the feedbacks and also questions to help us improve the paper. So the paper here is mainly focused on the brain-derived EV separation method. We evaluated the influence and process parameters on the recovery and content of brain tissue-derived EVs. Let's start from the background. So as we already know, EVA is involved in wild regulating pathways in body by serving as messengers to regulate cell-to-cell communication. Also in the CNS systems, EVA will also found segregated from different type of CNS cells like the neuron cell, the astrocyte, microglia, and so on. So this part of EVA, they can carry both the disease-associated agent like the tau protein and the amyloid, or some anti-inflammatory factors to regulate the CNS disorder. Thus, to explore the composition of CNS-derived EVs may help us to understand the mechanisms of neurological disorders in the brain. So the goal here in this paper is to separate EV from interstitial spaces of the brain to get the CNS origin EVs. That means we want to get the first round of EV from the tissue before they leave brains and go to the peripheral systems. So the interstitial spaces include both the interstitial fluid and the extracellular matrix. We know that the extracellular space as support of surround cells. That means compared to the normal EV isolation from cell culture medium and biofluids, here we also need to be gentle to deal with the tissues to try to keep the cell integrity. Or if the cells were largely broken, then we may get some of co-isolation of intracellular vesicles with the same physical property as EVs. So this is meaningful work because this is the way if you want to know more about the cell or tissue specific EVs. So here is the list for some already published paper related to tissue EV separations. At present, we don't have a standard for the EV tissue separation, but we usually will start with separating the tissue cell with the fluid. Most of groups, at least here, they use different enzymes to help with dissociation and also combining with some mechanical force. But some of the groups, they treat the tissues generally, they usually just general cut the tissue, while some of the groups, they did the ground or minced. So these steps actually will lead to non-EV contaminants and also will challenge the definition of tissue extracellular vesicles. The key techniques most of groups use are sucrose density gradient and ultracentrifugation after they got their EV after they got their EV population. Almost all the group, they also look at the EV marker protein expressions. But here we found out not all the group, they also try to check the depleted markers, which can use to evaluate the non-EV contaminations exist in the brain tissue EVs. So here I want to remind the viewer of the MySAP 2080 again. So this is the this minimum information of starting of EV 2018 was published by the whole SF community, and I believe most of people in EV field may have read this. But here, what I want to emphasize again is the requirement for the EV content characterization. So first thing, we need to demonstrate the presence of the EV markers, both the transmembrane one and the cytosolic one. And except the two types of EV markers, the co-isolated expected contaminant should also be included to evaluate the sample purity. Like if we are doing the plasma samples, there will be abundant lipoprotein and albumin existing, so it will be good to also evaluate how much these things contain in your EV separation. And there is no specific recommendation on the universal negative or positive markers, but here for the tissue, again, like I mentioned, the cell made disruption while we dissociated the tissue. So here it will be included, it will be better for us to include some intracellular markers to evaluate the cell integrity while we're doing the separation. especially for the brain tissue samples. So I want to emphasize again, the purity measurement is critical for the brain tissue separation evaluation. So based on the recommendation from the MySAF 2018, we will highly recommend one study published in 2017 from one of our collaborators. So in this paper, they use density gradient UC for the EV separation from the brain tissue. And they performed a careful and extensive characterization to look at the enriched and depleted EV component in their preparation. This method was shown to be highly effective at eliminating cellular contamination that do not have the same density as EVs. So in this method, they generally got the tissue and then slice the tissue and digest them in the collagen S3. And then by the differential centrifuge, they separate the tissue cells debris with the tissue fluid, and then they load the 10K supernatant onto a triple sucrose cushion to further separate the EV. So I think this method actually is a great and effective method for purifying tissue EVs, but also the density gradient UC step is relatively time consuming. We need to make the cushion manually. Also, we need to wait for a long time for the centrifuge to separate those fractions by the density. So here, in this study, we wonder if the size exclusion chromatography can work as a substitute method for the density gradient, because the SCC method is faster. It only takes 15 minutes for the samples on the column, and also it's automatable. We can have some small instrument to control the drops, and so here we want to compare the SCC method with the published density gradient UC method, and to look at if the SCC method can also achieve the acceptable EV purity from the brain tissue. And also we wonder the different species, if there were any difference between the different species. In this study, we include both the human, mouse, and macaque brain tissue to look at the EV characterization from them to see if they have different effect from the same protocol. And also we evaluated if the pre-processing protocol will affect the brain-derived EV quality. Here we include the comparison between perfused and non-perfused brain, and also the brain sample with different interval time before process. And then finally, we also looked at the small RNA and protein characterization of a brain-derived EV and their source tissues to reveal the difference between tissue and their vesicles, and also to reveal the CNS cell origins of vesicles. So here is our method comparison flowchart. So basically, the first part, the brain tissue preparation, we got the digestion protocol from Dr. Willa and Dr. Hill group, and we did minor modification, include adding the filtration step here to further deplete all the cell debris between the 10K. And then after the 10K spin, we also collected the 10K pellet, which we named them as large EV, which based the separation method is lower centrifuge, but because we have added the filtration step here, so actually here in the large EV group, we didn't see a lot of super large things. We just want to include this group here to compare with the final small EV product, which went through further purification process. So after we got the 10K supernatant, the first method we used is the method which I mentioned just now, published in 2017. We loaded the 10K supernatant onto the SDGU, and then we onto these three triple sucrose Cushing, and then we collect different fraction, and then we use UC to wash those fraction to get the final small EV preparation. And another way we did is we load the 10K supernatant onto the size exclusion column. And also for this method, we will also get different fraction, and then we use either the ultrafiltration or the ultrafiltration to wash and concentrate our samples. To summarize here, the method we include into the comparison is the SDGU method, which published in 2017, and then the SEC plus UC wash and SEC plus UF. So like I mentioned, we got different fractions from both the SDGU and SEC. So first thing we look at is which of these fractions contain more vesicles we were interested in. So to do that, we collect the different fractions from the SDGU and SEC, and we tested the protocol on both the mouse and human tissues. And we evaluated the first thing is the particle yield per 100 milligram tissues. And this particle count was determined by the nanoparticle tracking analysis. Here we can see the particle yield was highest for the fraction 2 from the SDGU method and fraction 7 to 10 from the SEC plus UC method. And the particle concentration was below the reliable range of the measurement in the fraction 7 to 6 and fraction 11 to 14 from the SEC method. And by the TM, we look at the vesicle morphology. You can see in all of these fractions, there was some vesicle structure existed. And then we use the Western to look at the presence of EV-enriched membrane and cytosolic markers and expected EV-depleted cellular markers. So for the human preparation, first you can see there were abundant CD63 and CD81 were observed in the fraction and fraction 2 and 3 from the SDGU and fraction 7 to 10 from the SEC. And the fraction 11-14, we still see some CD63, but without CD81. And then compared to the small EV group, our large EV, we can see those parts of vesicles, they had lower but still detectable CD63 and CD81 too. If we look at the mouse isolation here, we can see the similar thing, the EV preparation enriched for some EV markers, and for the fraction 2 from the SDGU, we also saw the CD9 expressed here. Then if we look at the intracellular markers in the human, we can see generally we didn't see detectable connexin or the GM130 existing in our EV preparation compared with brain tissues. However, for the mouse brain-derived EV, you can see those parts of preparation retain some amount of cellular markers, include GM130, connectin, and BAPE, regardless of what separation technique we use. So based on the result above, we focused on the fraction 2 from the SDGU and fraction 7 to 10 from the SEC in further and compared the three methods I mentioned before. So here is the method comparison result. The first comparison is did on the human brain-derived EV. And here is the three protocol I mentioned, the SDGU, SEC plus UC, and SEC plus UF. And we still did the particle concentration. We use BCA, did the protein concentration, and also we calculated the ratio of particle to protein to use an indicator to evaluate the purity of samples. So here we can see the NTA suggested that the SEC method seems to yield a slightly higher number of particles as compared to the SDGU, and the protein concentration is pretty similar. So when looking at the ratio of particle to protein, the SEC method also result a higher ratio compared to the SDGU. And when look at the large EV, this group with lower particle numbers but highest protein concentration, which contributed to the lowest particle to protein ratio, which is also makes sense because this is just some crude pellet from the t-shirt, and these fractions are after the photo purification. And then we also try to look at the vesicle morphology. The similar thing, we can see both of these fractions are full of vesicular structure here. So then we try to look at the EV and the cellular markers inside our preparation. The first thing is when look at the SEC plus UC method, we can see this method can produce EV with similar purity compared to the SDGU, which means there is no connecting existed. But for the SEC plus UF method, we can see there is a small amount of connecting was found, which indicates the UC may have a slightly advantage in increasing the purity compared with the ultrafiltration method. And then we also applied this protocol on the mouse to look at the mouse brain-derived EV. But since our preliminary result indicated there were more contaminations inside the mouse BDEV with cellular markers, so here we also added the false method, which we only test once, but it's combining SEC with the SDGU. So we didn't see significant statistical result here between different groups, but when look at the SEC plus UF group, you can still see it will produce more particles compared to other group, and also have a higher ratio of particle to protein ratio. But then if we look at the TM result here, actually in the SEC plus UF group, maybe it's hard to see because the figure is too small. But I would say they look at the background, there were a lot of non-vascular background also exists in this group, which also suggesting the UF may retain more vesicles, but may also lead to more non-EV contaminations. And then we also try to do the Western to look at all the cellular markers. However, we can see the cellular markers GM130, connectin, and BIP, they were found in all the EV preparations despite the method. So based on the result above, we think the SEC method can achieve similar separation efficiency compared to the SDGU, and these two protocols, they both work well for the human, but they can deplete all the cell contamination in the mouse vesicle preparation. So in the second part of the paper, we're trying to look at the pre-processing factor if them will affect the BDEV quality. So the factor we detected here include the PBS perfusion or without perfusion, and the post-mortem interval time, which here is the time between the animal death to the sample processing. So the first part is the effect of PBS perfusion on the BDEV separation. So in the animal model, it's often possible for us to choose if we want to perfuse or do not perfuse the animal with buffers such as PBS at necropsy. So here we compare the perfused tissue with non-perfused tissues. The first thing is there is no remarkable difference was observed for particle or the protein yield between perfused and non-perfused tissue. But if we look at the Western blot, we can see there is the perfused can reduce the amount of GM130 in the mouse brain-derived EV compared to the non-perfused, and however, the BIP and the connexin looks just similar. Also, some of the EV-associated protein like the TSG1, O1, and the syntanin, they were also lower in the perfused group compared to the non-perfused. So here, the reason may be the PBS can flush of the brain cell debris and also some blood cell derived vesicles. So we feel like maybe the PBS perfusion will be recommended for the animal models if we want to separate more clean samples from the tissue. And then we also try to look at the effect of postterm interval on the brain-derived EV separation. The reason is when the brain tissue from the animal model can always be processed immediately after necropsy, the human brains we acquired from the brain bank usually after varying time for the storage and different temperature. So to assess the influence of the length of time between after brain sectioning and the final BDV extraction, we obtained the occipital lobe of macaques, and we we divided the lobe into 3 parts and put them into the room temperature for 2 hours, 6 hours, and 24 hours. And then we use SEC plus user method to purify the EV and to compare the final output. So here, this is the particle count result. The first thing we can see is from the small EV group, it seems brain stored for 24 hours can produce more vesicles compared to the 2 hours and 6 hours. But when we look at the large EV group, there is no significant difference between these 3 different time points. And the PM here, you can also see plenty of particles here. The morphology is not different compared to the things we saw from the human and the mouse, and also there is no obvious difference between these different time points. And then we try to look at if the post-mortem interval will affect the brain-derived EV RNA content. So in these figures, we only include the 2-hour and 6-hour result. That is because when we look at the quality control data of the library prepared from the 24-hour samples, we saw a lot of degradation, so we didn't sequencing it. So here, when compare the 2-hour with the 6-hour, we can also see the library size. The mapped read count is lower in the 6 hours compared to the 2 hours in both the small EV and large EV group. When look at the RNA byteps, there is minimal RNA byteps difference between different time points. But if we look at the microRNA and pre-microRNA ratio, there the pre-microRNA and microRNA is slightly lower in the 6-hour samples compared to the 2 hours. Also, what I want to mention here is when we look at the small EV and the large EV, we can see a strikingly different proportion of RNA byteps between these 2 vesicle subtypes. And then we try to look at the microRNA diversity in the 2-hour and 6-hour samples. We filter the data to choose the microRNA with row count higher than 5. And then we can see in both the small EV and the large EV, the microRNA number detected in the 6 hours is lower than the 2 hours. And all this data, the mapped risk counts decrease and microRNA diversity decrease. Those are all perhaps due to the degradation along the time. And then we also tried to look at the effect of postmortem interval on the brain-derived EV protein content. But here, unfortunately, we only got limited number of protein were identified in the small EV and the large EV. This may because of we input less of protein here. We only use 1 microgram to do the profile. But anyway, when we look at the numbers here, the numbers of identified protein increase in the small EV and large EV with higher, with greater PMI, while the BH sample, the number in BH sample appear to decrease in the BH. So though we still, so when we get limited number of protein, we still try to do the geo enrichment to look at the cellular compartment may match to the small EV and large EV content. So here we can see the protein identified in the small EV group will especially enrich the full non-EV, the lysosome and the membrane associated terms. And we can see here is a stronger enrichment of cytoplasmic component at the 24 hours, which might also indicate more cell disruptions in the 24-hour samples. And when we look at the large EV, this group shared many components with the small EV, include the exosome and the lysosome fraction, but also it enriched for some terms such as the decimosome and the centrosome, which didn't appear in the small EV. So in summary, we think the longer PMI may lead to the tissue degradation and the cell broken. So for the animal samples, we will recommend to process and frozen the samples as soon as possible after the necropsy. So in the last part of the paper, we also try to look at the small RNA and protein content of both the large EV and the small EV, which separated by the SEC plus IUC method, which we modified. The first part is the small RNA profile data of BDEV from the macaque and the human brain tissues. So the first thing is we compare the RNA beta-types between different groups. Like I mentioned before, in this macaque data, so we can see the small EV and large EV with distinctly different RNA beta-type distribution. And we can see the similar things here in the human brain-derived EV. The microRNA, small nuclear RNA seems more enriched in the BH, while the tRNA fragment were more enriched in the large EV, and they were rRNA and the mRNA fragment exist in the small EV. So the result we see here is pretty similar to the macaque result. And then we also try to do the multi-dimensional scaling to show the pattern of small RNA expression between the BH, large EV, and the small EV. Again, we can see the pattern, the small RNA expression pattern was different not only between the BH and the EV fraction, but also between the small EV and the large EV group, group in both the human and the macaque. And other things, the variance in the small EV seems larger than the large EV and the BH, which may be caused by the further complicated purification steps. And then we moved to look at the microRNA diversity again in the macaque brain samples. Here we can see there were more microRNA identified in the large EV groups compared to the small EV groups. And then we further look at this 41 protein overlapped expressed in the large EV and the small EV. Among these 41, there were 28 microRNA, they were differentially abundant between the small EV and large EV. And here is the heat map for the result. We have two large EV group and two small EV group. You can see among these 28 proteins, some 28 microRNAs, sorry. Some of microRNA, they were more enriched in the small EV, while some of them were more enriched in the large EV. And if we move to the human data, it's also similar. When we look at the microRNA diversity, it's highest in the brain homogeneous and then large EV, and then the last one, the small EV. Again, if we look at this overlap the thing between these three groups, among this 180 microRNA, there were 49 differentially abundant microRNA between the brain homogeneous and the small EV group, and the fold change higher than 2. And this is the heat map here. You can see this cluster 1 microRNA, they were highly enriched in the BH, while the class 2 microRNA, they were highly enriched in the large EV and the small EV group. And also, if we look at the large EV, it more seems like it's intermediate between the BH and the small EV. So collectively, I think this data shows that large EV and small EV, despite some likely overlap, may have a different proportion of small RNA and including the microRNA. And this is also a proof that not all the small RNA are uniformly loaded from the tissue cells into EVs. And we also identified a small number of microRNA. They all appeared to be more enriched in the small EV compared to the large EV or the BH. And this result is with consistency across the two investigated species. So I think for the investigation on how this microRNA loaded in the vesicles would be interesting to look at. And then we also try to look at the protein profile of brain-derived EV from the human brain tissue. So this part I didn't include the macaque data because, like I mentioned, there is only a limited number detected from the macaque. But the human, for the human data here, we can see here is the identified protein number in the small EV and the large EV. We can see there were 167 protein were overlapped detected between these two groups, and the percent is around 40%. And there were more protein were only detected in the small EV, and also small amount of the protein were only detected in the large EV. When we try to look at the detected proteome and match them to the top 100 protein existed in EV marker database, include the vesicopedium, the EVpedium, and the exocard. We can see here in the small EV group, there were 71 proteins can match to the top 100 protein in the EV database, and in the large EV group, there were 65 proteins, which for the proved our preparation showed the protein content should match to what have been reported in the EV database. And in the previous slide, I have shown that the Western blot result on both the extracellular markers and the cellular markers. But for the Western, you need to input a lot of samples. So we only did a few of protein by the Western, and here from the proteomics data, we dug out more markers and plot the result here. So for the extracellular vesicle proteins, you can see they were all found in all types of samples in both the BH, the large EV, and the small EV groups. But certain EV markers was only observed for the small EV, included here the two tetraspanin, CD81 and CD9, and also these two cytosolic protein floating 1 and 2. They were only enriched in the small EV, but not in the large EV. And the annexin family seems can be detected in both the small and large EV, but there were two subtypes, the A3 and A4, they were only showed in the small EV. And also, if we look at the cellular markers, you can see most of these proteins, they were mostly enriched in the BH, including those associated with the mitochondria, the ribosome, and the nucleus, and only small amount of this protein were found in the BDEV preparation. If we look at the nucleus marker, you can see most of these nucleus markers, they were found mostly in the large EV preparation, while there were only a few that were found associated with the small EV group. So overall, I think the proteomics data we put out here is consistent with our Western result and shows the EV markers enriched in our vesicle preparation, while the cellular markers enriched in our brain homogenein. And then we also try to look at the CNS markers expressed on our brain-derived EVs. So we found that BDEV also carried markers of central nervous system cells types, include the neuron biomarker, the macroglia, oligodendrocyte, and the astrocyte. And we already know some of the groups, they try to use L1 chem to capture the neuron-derived EV in the blood. So here, I think from the result here, it's promising for us to develop a panel which can use to capture different CNS cell origins EVs in the periphery samples. And I also tried to do the geo-ontology analysis by both the string and the font-rich software to determine the cellular compartment of protein recovered from the BH and the BDV preparation. So here we can see a large portion of protein associated with both the large EV and the small EV. They were enriched for the EV-related terms like the vesicles, the cytoplasmic vesicles, and the exosome, and so on. And the terms, the membrane and the plasma membrane, the cell periphery, they were only enriched for the small EV group, but not for the large EV and the BH. Also, while the nucleosome and the chromatin proteins, they were only enriched in the BH and the large EV, but not in our small EV preparation. So I think, again, this supports our conclusion that the EV content is different from the origin tissues and also more extracellular compartment enriched for the EV compared to the BH. And then we also try to do the pathway prediction by both the KGG and the record time software to predict the EV to predict the brain-derived EV function. And you can see there is some CNS-related function include the synaptic vesicle cycle, the axon guidance, the neuron system, and the L1 chem interaction. But also there were some other identified process related to the metabolism, endocytosis, the immune system, and the memory trafficking, and so on. So this result may indicate the function loading of this protein into the brain-derived EVs. So here I want to summarize our conclusion again here. So the first thing we found is there were species difference reacted to the same protocol, like the human brain, the human preparation are always cleaner, while the mouse and the macaque we always saw some kind of intracellular contamination. This may because of the tissue structure difference. All those tissues have different react sensitivity to the enzyme, but the clear reasons still need to be determined. Also, we found the tissue pre-processing parameters are critical, and we will recommend perfuse the sample if possible, and also minimize the PMI for the animal models. And then we also found the protein and RNA composition of the brain-derived EV displayed several differences compared with their source tissues, and also the brain-derived EV carry some CNS cells markers, and also some molecules may relate to the neurological functions. And I also want to talk about our study limitation. So the first thing is for the conclusion come from the method comparison. We couldn't use exactly the same piece of digested brain to compare the different methods, because for each of the individual, we only get limited amount of brain tissues. So there maybe will be some variance between different tissues and will affect the conclusion. And also for the comparison between 3 different species, because of the availability, we actually use a different brain region for these 3 species. And I summarize some difference here. So for the human sample, we use the parietal cortex, which is the gray matter only. But for the macaque sample, we got the occipital lobe, and those are lobes with both the gray and white matter, because you know the macaque brain is smaller and it's hard to totally separate the gray and the white matter. And then for the mouse, it's a smaller brain, so we took the hemisphere of the mouse without the cerebellum for the purification. And also another variance here is the post-mortem interval. So for the mouse brain, we took the brain and immediately snap frozen them or process them. So the postmortem interval here is 0. For the macaque, we got the tissue from our retrieval lab, so it usually take 2 hours for us to get the sample after the necropsy. But for the human sample, what I want to mention here is we got the human sample from the brain bank. So the postmortem interval actually varied a lot from 3 to 21 hours. But like the data showed, it's still you can still see the human preparation can produce relatively clean EV compared to other tissues, while for the macaque brain, when we put the tissue for 6 hours or 24 hours, there will be a lot of degradation. So I think this is also a difference between the human and macaque species. And also, I put the particle yield per 100 milligram tissue and the protein yield here for you to add the reference. But again, they are from the different regions. But for the particle yield, we can see the human brain seems to produce highest particle yield, while the macaque is the lowest and the mouse is the moderate one. But if we look at the protein yield, you can see here the mouse, though the particle yield is not so high, but the protein yield is the highest one compared to the human and the macaque, which is also consistent with what we saw from the Western, like there were more intracellular contamination exist in the mouse brain preparation. And another thing is for this protocol, we add the filtration steps to strictly eliminate the cell debris, but this step will also eliminate some larger EVs. So this protocol will need to be optimized based on people's following study and different study purpose. And also, we saw the lower coverage for the macaque brain-derived EV proteomics data, maybe because of our lower input of the protein. And then all this data come from here. They are based on the profile and the bioinformatic analysis. So the further verification and the functional study will need to be followed to verify the findings here. So yeah, I think that's I have for this study.
Great. Well, thanks very much, Eyal. That was I really am appreciative that you took the time to share that with us today. And it looks as if we have a few questions. So, Eyal, could you perhaps stop sharing your screen now? And we'll see if we can get some interaction going here. So currently everybody is muted. And I'm going to unmute specific people. And if you need to talk, feel free to unmute yourself as well. And I just ask that you please keep yourself muted when you are not talking, because we have a lot of people in the call. So let me see here. There we go. All right, so I think we're probably not going to be able to get to all of the questions, but we have a few that have been submitted. So let's start with Lilia. Are you there? I'm going to unmute you here, and you can ask your question. Is this working? Okay.
Yeah, I hope, can you hear me?
Yes, yes,
Yeah,
loud and
hello.
clear.
Yeah, thank you for inviting. I really appreciated being a part of the discussion. I think it's really good, so to start with. And I was just wanted to ask not as like not a question, is still it's under the discussion that everybody knows that when we're talking about the large vesicles, we have to at some point they consider the apoptotic bodies, contamination, and it's just an idea which came to my mind when I seen that the number of the particles which were measured by NCA after 2, 6, and 24 hours were constant for the large vesicles. And well, you also got the annexin 5 and the rest of the annexins as well, the expression in the proteomics. And I'm just like wondering if you could just comment a little bit how their mechanism of the necrosis and apoptosis is going in this tissue, and does it really make sense to talk about the apoptotic bodies contamination or not? I mean, that would be interesting to me as well, because basically I'm trying to apply more or less the same protocol for the purification and visualization, but instead of the cross I use yodixonal density gradient. So yeah, could you yeah, just like say some couple of words, what do you think about the potential ability to have an apoptotic body contamination in your large vesicle preparation? It doesn't really make sense to talk about it at all in your case, or
So that's a good question, and I think I'm going to let Yao answer it. But I just wanted to note that there was also another question about the size of the filters that were used for that pre-filtration step, and so we took a very stringent approach to that in trying to do a very gentle filtration. But we were using 200 nanometer filters. So when we say large EVs, they're actually pretty small, and certainly smaller than a lot of the definitely the larger the larger apoptotic bodies, I should say. So yeah, I'll hand it over to you.
sorry. Yeah, I mean, I understand that you got this filter and you use it for the pre-filter and the sample makes sense, but well, know that their membrane is pretty flexible, and it also could be just a little chance of having something passing through the
Yeah,
filter.
certainly. So Yao, do you have any comments on looking for apoptotic bodies and their fragments?
Okay, sorry, I just lost my connection, so I don't even know if I finished my slide.
Yeah, you finished.
Oh yes.
Okay.
Oh yeah, everything was good. Yeah, thank you. Sorry,
Yeah, because it just interrupt and I
I didn't realize that you weren't there. Yeah. So anyway, Lilia
sorry.
was asking about the about apoptotic bodies, and then I explained a little bit about the filters and how it was a very stringent. I mean, it was a very restrictive filtration step that we did. So we were just wondering if you had any comments about apoptotic detecting apoptotic bodies.
I mean, we do not look at the apoptotic markers, but then other thing I wonder is what's the size for the apoptotic bodies? Is that larger or
Yeah. Well, so
Yeah,
I
well,
mean,
yeah?
I guess classically, it's interesting that classically the apoptotic body is usually said to be larger than 1 micron, but
Mm-hmm.
it's clear that I think now there's actually a paper in JEV, I think, on apoptotic bodies and how they can be much smaller than that too. I mean, so there are products of the apoptosis that are going to be smaller than a micron, and we don't want to exclude them just because they're smaller. So that was work I think that from Ivan Poon's group and Amy
yeah.
Baxter um, in Melbourne.
Yeah, sorry, can you just clarify? I don't know whether the size of the cells, if that's just smaller, it could cost you there some apoptotic bodies in the preparation, just in case. And uh, do you think that they might be there at some point, and that's why they're functionally not really, interested anymore in analysis the proteomics and RNA and so on. It's just like question.
Yeah, I think that's something that maybe yeah, maybe we can even examine that using the proteomics. Right. I mean, if we didn't see anything that jumped out at us, we could still go back in and look at that data set. So yeah, well, thanks very much for the question. I'm going to move on to Nikki. Nikki, you had a question about the technical versus biological replicates.
Oh, yes. I just was wondering, I think you answered it later on when you showed that it was different individuals and different animals, for example, for each of the replicates that you did.
Yeah, did you get that? The question is whether the replicates were technical or biological.
For the animal experiment, or for which part?
Yes, the animals and the donors. So the human donors and the
so the human donors in this project, we include 7 human donors. So all the proteomics and the sequence data, they will come from 7 donors. And for the animals, it kind of varied, but for the post-mortem interval, we have 4 animals, but we use 2 of them to do the Western and some general EV characterization, and then another 2 for the proteomics and the sequence. But we did see the consistent result between those 2 used for the profile.
Okay, thank you. Great study.
Thank you.
All right, let's move now to Clayton, who asks, when comparing the proteomics, why do we see more PM markers in the small than the larger EVs? Clayton, I just unmuted you if you want to ask that.
Hi, Ken and Yao. Can you hear me?
Yes.
Yeah,
Okay.
I can hear you.
Yeah, thank you for having everyone. This was very nice and very nice work. I saw that in the 2 analysis for the proteomics that plasma membrane proteins jump out in the small EVs, but actually not the large EVs. And is that a little counterintuitive to what we might expect there, or do you have a reason for that?
Yeah, were you able to hear that? Sorry, you might want to start again because I think you were frozen there for a minute.
It's frozen out screen. Okay, so when we compared the proteomics data in the fun-rich and the later slide, in the very first proteomics slide, we see that plasma membrane proteins jump out in the small EVs, but not the large. Is that a little counterintuitive, where we think that microvesicles and apoptotic bodies generally come from budding?
So are you talking about this slide?
Yeah, so over there in the right-hand chart, the plasma membrane and membrane proteins are really prevalent in the small EVs.
Yeah, they are primarily in the small EVs. So we assume like the small EV group, they are not with the solely origin, like they may come from the multivesicle body, but also the plasma membrane. But for the large EV, to be honest, we named them the EV, but actually from our characterization, it's an intermediate things between the BH and the small EV, and also you can see a lot of more contamination exists in the large EV, and they will only come from basically the 10K palette. So I think the large EV group may more come from the tissue and also with some extracellular thing, while the small EV, they may contain different subtype like the macrovesicle or the exosomes.
Okay, so that may be included in the microvesicles are in the small EVs. Okay.
Yeah, that's maybe the reason.
Thank you.
Thanks for that question, Clayton. And then we had a question from Yvonne about L1 cam, and then we also got a response about that from Raha, I believe. Is your question answered, Yvonne, or did you have something else that you'd like to follow up with? I can't seem to unmute you. Come on now.
So to clarify, I've seen
There you
him
are.
present on this, but I wasn't able to find a publication. So maybe it's not, or maybe it's not its own and I just couldn't find it.
Yeah, so I mean, I think the question here is that question about L1 cam, and so you know we see L1 cam definitely in these brain-derived EVs, and this is what Dimitrios Kapagiannis and Ed Gutzel and others that they've collaborated with have used to try to obtain those vesicles from blood products, for example. And the question was, is L1 cam really specific to neurons? And I think, I don't know if Dimitrios is on the call here, but I think he would tell us that no, it's not just neurons. And it's also not just central nervous system neurons. Of course, you can have neurons in the periphery too. So it does seem to be a marker that his group has used to great success and kind of determining some markers of early cognitive decline, later cognitive decline. But it is not strictly on neurons. And I think one of the things about this study and other studies like it is that we can identify additional markers. And now that we have the ability to do some single particle phenotyping, we can, at least potentially be able to look at multiple markers. And that would help us in understanding subtypes, different kinds of neurons that these cells might have come from. So I think it's important to look at the proteomics here and maybe use that to guide our continuing search for these brain-derived EVs in other compartments. So we have, I want to keep this to 1 hour. So unfortunately, we would not be able to get to the rest of the questions right now, but this is online. And so we can follow up, and we will follow up with those of you who ask questions, and we'll probably post a summary. We have also recorded this, so I hope the recording worked. I hope you're not mostly looking at my cat or something by mistake, but we're going to post the recording likely on YouTube if it worked out. So thank you again, everybody, for joining. It's been really good to interact with you today, and I hope that this resource and other resources that are being developed will help to keep our community strong and moving forward, even while many of us are stuck at home. So do reach out if we can help in any way, and let us know if you have some papers or some results that you really want to share in this format. We'd be glad to work with you. So have a great day, morning, evening, wherever you are, and look forward to talking again soon. Bye, everyone.
Thank you. Bye.